In 2001, a senator and a TV presenter in the United States received anonymous mail. The letters contained a high concentration of anthrax spores. The Senate, media organizations, and government buildings were all affected. Entire buildings were repeatedly evacuated. Hundreds of people were treated with antibiotics, and 5 people died as a result of inhaling the deadly anthrax spores. These anthrax attacks also had an impact in Switzerland. In 2005, postal workers in the canton of Zug discovered letters containing a white powder. This was the prelude to a series of threatening letters sent to different parts of the country. Terrorists are not the only source of biological threats. In 2003, the Swiss Federal Office of Public Health banned Asian exhibitors from the watch and jewelry fair Basel World. The reason? Severe acute respiratory syndrome, or SARS for short. The first cases of SARS were found in China at the end of 2002. 1 year later, SARS infections were identified in over 30 countries, including Switzerland. Globally, around 800 people died as a result of a SARS infection. Another example of an infectious disease with the potential to cause an epidemic is bird flu. A package containing the suspicious powder is brought to the Spiez laboratory. The facilities where suspected bioterrorist substances are analyzed are located in a high-security building. Access is restricted exclusively to authorized personnel. The suspicious package is examined using a complex glove box facility. Great care is taken to open the package, and the contents are logged according to a predefined checklist. The use of these special inbuilt gloves means that laboratory staff are never directly exposed to potentially dangerous substances. A sample is taken from the suspicious powder and transferred to a culture medium. The sample is then incubated for at least 18 hours at 37 degrees centigrade. If the sample contains bacteria such as anthrax, easily observable bacterial colonies begin to grow from each individual bacterial cell. These colonies are then examined and identified. The identification process involves marking the bacteria with specific antibodies, which are tagged with a fluorescent dye. This makes it easier to observe the bacteria under the microscope. The Spiez laboratory can also identify viruses. Many different cell types are required to identify viruses. These are stored in liquid nitrogen at -192 centigrade until they are needed. Examples of the most dangerous pathogenic viruses are Ebola, smallpox, and influenza. Viruses are the smallest type of microorganism and require a living cell in order to reproduce. A live virus must first be grown in a cell culture before it can be identified in a sample. In addition, a sample which potentially contains a virus is assessed using a cell culture. Viruses destroy cells. Using their antennas, they adhere to the surface of the cell and penetrate the cell interior. There, the virus splits into its individual components, and its genetic information then can penetrate the nucleus of the cell. The DNA of the virus can replicate itself here many times over before being discharged again. Many new viruses can be created from one single virus and can go on to infect new cells. In most cases, virus replication causes cell damage. As these are visible under a light microscope, the experts from the Spiez laboratory are thus able to assess the extent of the damage. Using antibodies tagged with a dye, the infected cells become stained, enabling the precise identification of the virus. While it is impossible in many cases to identify the viruses themselves, the antibodies which are produced can be identified. In addition to these traditional virological methods, the Spiez laboratory also uses molecular biological procedures for virus identification. These allow the specialists to unravel the DNA of the virus and identify bacteria. First, the viral DNA is extracted and is then reproduced using a highly sophisticated procedure. These procedures are extremely sensitive, and all precautions should be taken to ensure there are no impurities. The replicated DNA can be identified using fluorescent-based techniques. The resulting curves provide information on the type and quantity of the given pathogen. The biology division of the Spiez laboratory also have the facilities and know-how to handle toxins, such as ricin or botulin. Botulin is one of the most poisonous substances known to man. To identify toxins, the specialists use immunological procedures, similar to the procedures used to identify viruses and bacteria. They can also use mass spectrometry procedures specifically to identify toxins. These can ascertain the exact quantity of molecules, which allows the specialist to reliably identify the suspicious substance. The identification of bacteria, viruses, and toxins is also very important for the military. In the 1990s, Iraq was discovered to be developing a terrifying bioweapons program. Even today, the military deployment of bioweapons remains a possibility. For this reason, the Swiss Army is commissioned to make the necessary preparations for a possible bio threat. To this end, reserve soldiers receive the necessary specialist training in the Spiez laboratory. As a result, this military personnel could also be deployed to assist in a civilian incident. Given that the staff are organized along military lines, professional round-the-clock deployment can be organized in a very short space of time. The Spiez laboratory makes its expertise and know-how available to international organizations, particularly the World Health Organization and the UN Environmental Protection Agency, UNEP. Experts from the Spiez laboratory also take part in missions to regions suffering from the aftermath of conflicts. For example, Afghanistan has serious environmental problems, such as contaminated drinking water. In collaboration with its international partners, the Spiez laboratory carried out on-site inspections. This work makes an important contribution to reconstruction work and can open up new horizons for the Afghan people. Microbiological drinking water inspections are carried out by the Spiez laboratory on behalf of the Swiss Army. It checks that the quality of the drinking water complies with the legal provisions, thus ensuring that the Army has clean drinking water. The procedure involves filtering a sample of drinking water through a membrane filter by means of a vacuum pump. Since the size of the membrane pores is smaller than the diameter of the bacteria are retained in the filter. The next step is to carefully remove the filter and transfer the bacteria to a culture medium. During the incubation period, visible colonies grow from individual bacterial cells, and these can be counted using a magnifying glass.
You have a piece of equipment that they have to work with, and these are refrigerated.
The experts from the biology division of the Spiez laboratory actively contribute to international exchanges of specialist scientific knowledge.
This report also offers kits for most of the relevant bacteria and toxins.
They publish their results and discuss them at conferences with specialists from all over the world. The Spiez laboratory has an excellent reputation as one of the world's leading organizations specializing in NBC protection. Spiez laboratory experts actively work to ensure our safety. they are always prepared to investigate unknown suspicious samples swiftly and reliably. Their work covers a broad spectrum of possible threats, bacteria such as anthrax, as well as dangerous viruses and toxins. In addition, they carry out analyses of drinking water on behalf of its national and international clients. Where necessary, trained reserve soldiers can be drafted in to expand the laboratory's capacity. The experts in the biology division make an important contribution to making the vision of the Spiez laboratory a reality, namely a world without weapons of mass destruction.